This slide shows the other main group known as spirochetes. The top slide actually shows what's known as a vibrio, which is not classified as a spiral um, shape itself. It pretty much falls into its own classification for morphology and its characteristics of what we know as vibrio. Vibrio is part of the Vibrio cholera types of bacteria. Um, so we'll leave it at that. In the next two, we have the spirochete and the spirillum. The spirochetes are typically flexible, while the spirillum is typically a fixed or stiff spiral-shaped object. Here we have a summary of morphologies and cellular or cell arrangements. We have the three main characteristic morphologies: the straight rod, the coccus, and the spiral forms. And we also have a subcategory of the comma forms. The remainder of morphologies are typically known as pleomorphic. Arrangements are on the bottom, and we see a coccus or a bacillus in a diplococcal, a streptococcal chain. Coccus specifically in form of tetrad and clusters, whereas you don't typically find bacilli in tetrad or cluster formations. Let me briefly mention measurement. Measurement is measured in micrometers. This slide shows differences across small microbial organisms, from atoms to eukaryotic types of cells, and the prokaryotes fall in and around 1 micrometer. To give you an idea of what the organisms look like on culture, this slide shows a agar plate with bacterial cultures. Typically, we grow the bacteria in what we call a broth. We inoculate a broth, a cell culture, and then we plate it onto the agar as seen in the picture here. And we use a technique where we start out with a heavy growth, and then we slowly draw out those organisms until we get isolated individual colonies. Here's another example of a plating on agar of a bacterial organism. You can see this type of organism grows very thickly and tends to appear to be This slide shows the basic configuration of the cell walls of bacteria. On the left we have gram-positive, which is typically made up of an inner membrane and a peptidoglycan wall, while we have a gram-negative on the right, which typically has an inner membrane, a smaller cell wall, and then an outer membrane. When the bacteria are stained, the stain is taken up by the peptidoglycan cell wall, and the staining characteristics determine whether the organism is gram-positive or gram-negative. Here we have a basic gram stain slide with gram-positive rods on the left and gram-negative cocci on the right. As you'll note, typically the gram-positives will stain a darker purple color, while the gram-negatives will not take up the purple stain and typically stain a red color. Here we have an example of gram-positive staphylococci. You can see the spherical shaped organisms in a cluster arrangement that are staining a dark purple color. Here is an example of gram-positive streptococcus where you see chains of spherical bacteria staining a deeper purple color. This slide shows gram-positive streptobacilli. So we have the rod-shaped organisms in chains. In the background there appears to be some organisms that did not quite uptake the stain itself, or they appear to be possibly endospores, which we will discuss next. This slide shows gram-negative rods. They're a little bit difficult to see, but they are E. coli, which is a gram-negative rod. This slide shows a mixed number of organisms. We have some gram-positive staph here, and we also have some gram-negative, looks like diplococci, maybe perhaps gram-negative rods. Here it looks like a diplococci. Over here it looks like possibly rods. Here we have gram-negative diplococci. This is an example of Neisseria gonorrhea. Within these cells, these are actually human cells here, white blood cells they appear to be. We have, within the phagocytic cells, we have gram-negative diplococci in this area here. Here we have a mixed gram-positive in what we know as acid-fast, non-acid-fast bacteria in gram-positive clusters, probable staph. Here they say Staphylococcus epidermidis. Over here we have acid-fast, which is a specialized stain. In particular, we use it for looking for Mycobacterium. Mycobacterium tuberculosis is a pathogenic organism that causes tuberculosis in the lungs and other body tissues. Here is a sputum sample that demonstrates acid-fast bacteria in the red, in the background of cellular debris from a sputum. This would be typically done for patients suspected of having tuberculosis, taking sputum samples to look for evidence using acid-fast staining techniques. Here we have examples of using what we call malachite green staining technique to look for endospores. The spores themselves here where the arrows are pointing at, or the pointer, show that we have endospore formation amongst a background of rods. Typically these rods, although they're staining red in this photo, are gram-positive. Here is a slide showing malachite green staining technique to identify spore-forming organisms with the green stain. The other organisms that are still mixed in here are vegetative state, are bacteria that have not gone into an endospore forming situation.